Welcome to the UKCRF Network Phase 1 Framework Introduction. My name's Kim Lee. I'm the Senior QA Lead at the NIHR Southampton CRF. In this session, I'll be covering how it started, why the framework was created, implementing part of the framework, the risk stratification matrix and the associated contingency plans, and how it's been implemented at the Southampton CRF. This session is based on the session that was presented at the UKCRF Network Conference in 2022. On 15th of March 2006, 6 healthy young men in a Phase 1 clinical trial had organ failure after being dosed with IMP TGN 1412. The MHRA conducted an investigation into the conduct of the trial, and the investigation outcome shows that there were failure to complete the medical background of a trial subject in writing, and the medical history of one of the volunteers was not dated. 2 volunteers were allowed to leave the trial before it was confirmed that they had received the placebo. Parexel failed to review Genero's insurance policy, and later it was found did have an exclusion that would impact on the volunteer. There were no contract at the start of this trial. One of the doctors did not have adequate training or experience, and there were no 24-hour medical cover. Following the investigation, the MHRA created and formally implemented in April 2008 the MHRA Phase 1 accreditation scheme. This is a voluntary scheme. That means any sites can sign up to be part of the scheme. The scheme is a recommendation from the expert scientific group on Phase 1 clinical trials for organization conducting Phase 1 trials, in particular for those conducting first-in-human trials. The aim of the scheme is to make sure trials are as safe as possible and to create public confidence in regulation of Phase 1 clinical trials. To be accredited, organization in the scheme have to exceed basic regulatory good clinical practice, GCP, standards by having additional procedures that include the high standards for avoiding harm to trial subjects and for handling any medical emergencies. Since the implementation of the scheme, several CRFs in the network are accredited units or were, and had noted that the scheme is not suitable for public sector facilities. But most of the issues encountered by the CRF within the MHRA Phase 1 schemes are due to the ability to oversee all aspects of GCP rather than the Phase 1 scheme per se. What that means is that units under the scheme would also require to oversee the day-to-day running of the study. So when the study is being inspected under the scheme, the Phase 1 scheme, the report is actually split into GCP, your normal, your average inspection report, as well as Phase 1 report. When we're being inspected, we are not just being inspected whether we have implemented the Phase 1 aspect, the Phase 1 scheme aspect. The CRF concerns about site being responsible for regulating quality when sponsors control the data, specifically who should be acting as the policeman. So, as part of the scheme, all units under the scheme must have assurance that all data from participating sites have been appropriately reviewed and checked. And that's a big task because these sites could be worldwide. So how does a site under the scheme have the ability to assure that the site in America, their data has been reviewed? Currently, if a study is turned down by a CRF in the Phase 1 scheme, it doesn't prevent it from being conducted elsewhere, where they may not have a system to acknowledge the risk. To address some of the issues, a meeting was held between CRF directors on behalf of the UKCRF Network, the MHRA, and the Health Research Authorities, HRA. In the meeting, it was agreed that a framework was in the best interest of patient safety for facilitating highest risk early phase and experimental studies. It would have a similar approach to the MHRA Phase 1 scheme of risk assessing studies. It will ensure a contingency planning, so we all work the same across the UKCRF network, and it will focus on the highest risk study and the needs of the NHS. And it will keep UK competitive, so it's a very pragmatic approach to focus key resources on the highest risk trials. So the goal is to create a framework to ensure minimum standards for safe conduct of Phase 1 trials in the CRF NHS. So the UKCRF Network has created a framework document that outlines of areas of the formal MHRA scheme that should be adopted as best practice for all CRFs for the highest risk early phase and experimental medicine studies, underpinned by other resources and tools to be shared across the network. It describes a governance framework under which the highest risk studies should be managed. The network has also created an initial risk assessment to identify the highest risk studies. The current version has been designed so that all units can meet the common standard without additional investments. Also, basic data could be collected by all units regardless of whether or not the CRF is choosing to meet the formal MHRA standards. The purpose of this framework is to provide a guide to enhanced governance processes, including risk assessments that will allow all CRFs to work to a minimum agreed standards for the highest risk studies. What it is not yet is a formal MHRA accreditation or MHRA light scheme. Within the framework, there are 5 areas to consider for the highest risk trials. That is the clinical trial design and setup, medical emergencies and facilities, staff, subject recruitment identifications, and quality management or quality system. In this section or in this session, we're mainly going to be focusing on clinical trial design and setup. What this involves is a process in place, identifying the highest risk trials using the risk stratification matrix, understanding the associated contingency plans and dose escalation. Clinical trial design and setup. In the framework, it outlines a requirement for safety information availability and insurance. These are the 2 requirements that is in the framework, as you can see, which is ongoing safety and toxicology data updates to the CRF and local phase 1 immediately to ensure the safety of the subjects in these early phase trials. And the sponsor must also ensure appropriate indemnity insurance is in place. What you most likely find is that your local R&D and R&I would already have these agreements in place. Early phase committee, EPSC, or other local processes. In the framework, it outlines a committee or process to allow appropriate risk assessment of the phase 1 and experimental studies. The committee should have a wide range of expertise because you can have a wide range of phase 1 studies that requires assessments. What you might want to look at is there already a committee or committees that's in place at your local site that you may be able to join or may extend their remit to phase 1 studies? There is a term of reference in the appendix of the framework that you can use as a guide. Risk assessment, risk management, and mitigation. A formal risk assessment of phase 1 or highest risk experimental medicine studies to ensure appropriate contingency plan is in place to mitigate any known or possible risk. This needs to be reviewed by the EPSC and to ensure that you have captured all phase 1 studies or highest risk experimental medicine studies, you need to embed it within your local R&D or R&I processes. Risk stratification matrix. As you see on the right, our risk stratification matrix, which can be found on the UKCRF Network portal. It was developed as a tool to risk assess a phase 1 classified study and allow a pragmatic and risk-adapted approach to manage hazards associated with the trial. So from our experience at Southampton with assessing phase 1 trials, we've noted that not all phase 1 trials actually carries the same risk. So this tool really helps us to allocate the appropriate amount of resources to manage the risk that actually is presented. The outcome is a combination of the score from the status of the intervention, as you see going across at the top, and the trial's design or the study design going down at the bottom left. And if necessary, any additional factors such as study team experience with intervention, and the number of intervention, right at the bottom going across the matrix. It is the responsibility of the PI to make these assessments, and where necessary is peer review to ensure the outcome reflects the study conduct at their site. The outcome would be either red, orange, yellow, or green. This is based on the matrix that you would have used when reporting incident at your local trust. For this matrix, here's the key to what the outcome color means. Green means it's low risk, yellow is low medium risk, and orange is high medium risk, and red is high risk. Medium risk is split into low and high, will be explained later in the session. So what do we mean by the low, medium, high risk within this matrix? The MHRA Grey Guide has a really good definition of these risks to give clarity and another indication if the matrix outcome is appropriate. So for low risk, according to Grey Guide, it's no higher than the risk of the standard medical care. Medium risk, whether it's low or high medium risk, it's somewhat higher than the risk of the standard medical care. And high risk is remarkably higher than the risk of the standard medical care. Let's have administrated. So let's have a look at the definition of the categories that's in the intervention status. So the definition in the categories of the intervention status are from the EU Clinical Trial Regulation Guidance on assessing risk of a study. And it's really based on how much safety data or the level of safety data you know about that particular intervention. So starting from the left, this is considered to be low risk as the intervention is licensed, and we know lots of safety data about that. And as we move to the right, the risk increase because we have a reduced amount of knowledge about that particular intervention. The term intervention is used as this can be used for IMP, medical devices, ATEMP, and any other terms can be used for what we're doing to our participant. So let's have a look at the first category, used as per license or off-label use is established, practice and supported by published evidence and or guideline. So I believe this is quite straightforward, using it as it is, as licensed, or that you know it's safe to use for another condition because of the published evidence or guidance or guidelines. So for example, chemotherapy used for one type of cancer, and then it's used to treat another cancer, and it's been supported by guidance from organisations such as NICE. The next category we're looking at is licensed and used in new formulation or new delivery. So a licensed product that was licensed as an infusion, then changed to a capsule, or that it's been licensed to use as a single treatment, and this study is looking at in combination with another licensed intervention. So one example I have would be a standard of care chemotherapy to treat bladder cancer. In the study that came to us, the study was investigating if it's more effective if the chemo was administered directly to the bladder or the licensed method of infusion. The next category is licensed but used in new age group or disease. So this is your study where it was licensed and adults, and then they want to see, can it be they're investigating it in children, or it's been licensed for asthma, and they are now looking at as a treatment for lung cancer. So what's the difference between this category and the first one we looked at? So the difference is that in the first category, it's been established, it's an established practice to use in a different disease or age group. And it's supported by published evidence and or guidelines. In this category, you don't have the established practice or the published evidence or guidance. So next category is unlicensed, used in healthy volunteers, but first in patient group. So this is unlicensed. So we don't really know what it would do. We don't know that much safety data, possibly. The best for unlicensed product or interventions, it normally comes with an investigator brochure. And in the investigator brochure, or IB, locally known, there will be a section in the brochure titled Effects in Humans. It's usually section 5, usually. And this is where you can see or find out, well, what has this intervention been tested on, what groups of people? And in this category, it's normally have been tested in humans. Now, this group of humans could either be healthy volunteers, but it can also be a patient group, as some intervention would not be safe to be used outside that particular disease group. Again, if we're looking at things like cancer treatment, it would be unethical to use it on healthy volunteers. So they would use it within their own patient group. But essentially, this is an unlicensed product with information about the effects in humans, whether it's through ongoing trials or completed trials. And the very last category is unlicensed and never been used in humans, whether that's the healthy volunteer group or the patient group. And if we go back to the IB, under effects in humans, you'll see that there would be absolutely no data for it. So this is where, this is where it would fit in. So now we looked at the intervention statuses. We are now going to look at study design. Again, these categories are from the same EU clinical trial guidance as the intervention status. First one we're going to look at is dose escalation. So this dose escalation with multiple or complex cohorts, such as your single ascending dose and multiple ascending dose used in combination. You would have dose leaders, and the purpose is to establish recommended dose, double-blinded and randomized, or for ophthalmology, double masked. With these two design, I would just not take that into consideration. And that is because whilst both design carries risks, these have been already included in the contingency plan, so we don't need to take that into consideration. The next category we're looking at is dose escalation of a single dose. So there's only one cohort taking place at a time, which is different to the previous one because you would have the single ascending dose and the multiple ascending dose at the same time. So in this category, the dose escalation would be only one cohort taking place at a time. The single dose, this is for studies where the participant is only receiving one dose. There are no other cohorts. There's no changes to the dosage. Again, there is the category of single blinded or single masked. Same as before, you don't need to take that into consideration because the risk has been included in the contingency plan. Next category, your dose expansion or dose escalation with known or tested doses. The protocol will state if it's a dose expansion cohort, and it's something you would have discussed in feasibility as well if you're entering a dose expansion cohort. For studies where there is a dose escalation, and you've seen that these dosages has actually already been tested, again, go back to the IB, look into the effects in humans. If it's already been tested in ongoing or completed trials, this is where it would fit in. You'll see open label on here as well, same as previous. Don't need to take that into consideration. And the last study design category is dosage used as per authorization. So we're using it as it's been licensed to use. So once you have found the information you need, you will start running along the matrix. You would find a category that the study intervention fits in, as well as the study design, and then you run it along until you meet the square, and that is your outcome. You can leave it there, or you can run it along the additional risk. This is optional. It is at your discretion. If you do decide that you want to use it, you will start off where the risk outcome from the previous matrix. So previously it was orange, we'll start on the orange, and then you run it along like a risk scale. If you want to run it along multiple risks, you would start the next risk at where your last risk ends. So in this example, it ended with on the green, and then with the next risk, we start on the green and then we run it along again. Wherever you ended, that is your final risk outcome. So once you have your risk outcome, then you will select the appropriate associated contingency plan, which you can find in the worksheet tabs. So as you can see at the bottom, we've got low, medium, high risk tab. You select the one that's appropriate, and that would be your contingency plan. Contingency plans. The associated contingency plans outlines the additional process to implement based on the requirements from the NIHR Phase 1 accreditation. So we selected the additional processes going above and beyond GCP. The plans indicate if the additional process is required to be implemented according to the risk level identified from the risk stratification matrix. So to easily see the difference between all the three contingency plans, what I've done here is that I actually took each one and pop it all onto one page so we can easily see. So as you can see on the left, these are the additional processes and we have categorized them. And then on your right, you'll see lots of different colors, the green, the orange, and the red to indicate the different levels. Where you see cells that are white in the background, there's no color to them. These are the things that we have to have in place. So, emergency contact test. So for all study, regardless of the risk, we would need to perform an emergency contact test. For anything that's in the orange, that is where you can tailor to the study requirement. So the next one underneath contact test is the unblinding or unmasking system test. So you would only need to do this if your study has a blinded or masked design. Same goes with ICU notifications. You can also see it as identified by the PI. This is another way where you can tailor to the study according to what the PI deemed to be appropriate. So just going quickly back to the low or high, medium risk, that is your yellow and your orange. This is where it comes into play. If it's a low medium risk study, you may feel like actually for ICU notification, it isn't required. But for a high medium risk study, you might feel like actually we do want to notify ICU. So with the medium risk, you have much more ability to adapt to the study needs. Here is an example of how we use the risk stratification matrix in practice at Southampton CRF. At the top, going across, that is our R&D study setup process. And you can see at the bottom, this is the actual flow of the matrix and the CRF. So when a study has been identified and assessed, and when it's going through an arrangement, we will check if it's a phase 1 study, if it's been conducted in a CRF. Now, if it isn't conducted in a CRF, we won't go through any of this process. But if it is, then the PI will be asked to review the study against the risk stratification matrix and then complete the associated contingency plan and a dose escalation procedure form, if applicable, if it has a dose escalation design. The documents will then be sent to the EPSC for review. And if there's any queries from the EPSC regarding the study, then that will go back to the PI or sponsor, because some questions can only be answered by sponsor, to respond. And that will be sent back to the EPSC. And that cycle will go on until there are no other queries. And then the EPSC will issue the approval, which will be sent to our R&D, because our R&D, as part of their process, would only provide CNC once EPSC approval has been received for phase 1 study being conducted in the CRF. So this is how we embed the process and how we ensure that we captured all phase 1 studies running in the CRF, that they go through the risk stratification matrix and complete the associated contingency plan and, if applicable, dose escalation form. Ongoing review. Once a study has been reviewed and approved, it's important to have an ongoing review of the risk and contingency plan, as changes in a study once it has started may have impact on the contingency plan. It's important to have an oversight of protocol substantial amendment, staff changes, file notes, especially of safety events that had occurred, and monitoring reports and audit findings. At Southampton, the EPSC review notification and amendments as it comes through, and this process is embedded in the R&D process, very similar to at the start of the study when an amendment comes through. R&D would always ensure that EPSC is notified and will not issue CNC until they receive confirmation from EPSC that's been reviewed and there's no concerns or issues or impact on the contingency plan. Dose escalation. Going back to the flowchart that I've previously shown, you may have noticed that I've mentioned a dose escalation procedure form, if applicable. What that means is that for dose escalation, there is an additional process to have oversight of dose escalations. The following all have oversight of dose escalation. So we have the Data Monitoring Committee, DMC, or Safety Review Committee, SRC, or any form of committee, whichever title or acronym they use, have the responsibility to recommend the appropriate outcome based on the data set shown at point of dose escalation. The sponsor has responsibility to share that data that the DMC or SRC review and makes the final decision based on the DMC or SRC's recommendation. The PI has medical responsibility for the trial subjects under their care, ensuring that they have information about the data used, for example, the starting dose and for dose escalation decision, so they can make the decision for the subject under their care at their site. For more information, have a read of the blog post by the MHRA written by Jennifer Martin. The link will be in the description box below in the video. This is a fantastic blog post that has all the information about dose escalation and what it means in relation to the regulation. It has useful background information, a breakdown of what the regulation expects from the PI, the issue, and advice for site. So when it comes to dose escalation and the EPSC, in the framework, the role of EPSC is to ensure a copy of the full dose escalation decision documentation has been given to the PI for filing in the site file, and information of the QA, so the quality assurance, or the QC, the quality control, or SDV, source data verification of the data used to make the decision is available. Quite often in the dose escalation paperwork, that particular information is missing. And if you read the blog post, it does say within the regulation that the PI does need to be assured of the data has been quality controlled in order to make their decision at their site. So for example, where this information can be found, it can be in the full data set. It can be noted in meetings and presentations. If it's not in those documentations, then we would ask the sponsor to confirm by email. For less experienced researchers, the EPSC role is to ensure that local oversight of the dose escalation event that is formally overseen by the trial DMC. Dose escalation procedure. To find out what the procedure is for dose escalation in a particular study, this can be found in either a protocol or an SOP that's been issued by the sponsor or the CRO, a charter, or a study-specific plan. Because, from our experience, this information can be spread out over several documents, it might be easier to see the full process by collecting the information onto a separate form. At Southampton, we do have the dose escalation procedure form to collect this information, so we can just have one document, we can go to one document and have full view of what the process is. So as you can see on the right, that is our dose escalation procedure form. It requests information for the process of a dose escalation. So that is a series of actions or steps taken in order to achieve a particular end. And then we have information about the procedure for dose escalation, which is an established and official way of doing something. This really assists with identifying any concerns with the process that doesn't follow the MHRA guidance that is set out or explained in the blog post. It is really important to understand the data that's been used to make that dose escalation decision. Decision made on unvalidated data set, which means that data that hasn't been QC'd, QA'd, SDV'd, puts the patient and organization at risk. We aim for 100% validated of all data set. That is the gold standard, but it's not always possible, especially for studies where the patient is at risk of any delays when it comes to dose escalation. From our experience, it's really important to have an understanding of what is essential data to make that dose escalation decision and the percentage of that data. So it may not be 100%, it could be a risk-based monitoring program, especially if it's not a first-in-human study. This should all be communicated at study setup. For studies where it involves multi-centers, you should also have reassurance that the data from other participating sites have been validated. Another important thing to note is that the PI must be satisfied with and document their satisfaction with the sponsor's decision to dose escalate and the accuracy of the data on which the dose escalation decision is based on. PI responsibility, or they may delegate that responsibility to a member of the team, is to ensure the sponsored decision to dose escalate and the PI oversight of the data used to document in a site file. So that could be in the meeting minutes or meeting presentation. At Southampton, we also have another form to document when there is a dose escalation. And this form is called the dose escalation event form. And this is to confirm that the dose escalation conducted is as per the dose escalation procedure form. And it's a really great way to signpost to where we found this information and how we are sure that that decision has been made accordingly. In this session, we covered the purpose of the framework, which sets out the minimum standards for the CRF to implement. Doing so, this allows a collaborative or information sharing community across the network at a single trial level. It has a pragmatic and balanced approach as it identifies the risks for these individual studies and implement a proportionate process by really focusing the key resources on the highest risk trials. It also uses resources from the UKCRF Network and your local trust policies. It is also a streamlined process, which will really aid with the speed of the study setup. This concludes this session. There will be more sessions regarding the framework, either as part of the UKCRF summer school or training videos such as this one. Thank you.